Hey, good afternoon. You can hear me? Okay, perfect. So, today we have to speak about chemistry and biology together. For the practice sessions, since I'm having so much problems nowadays, like with my health, so I won't unfortunately do them with you, but I have questions which are not in the IMAT, since I think most of you or all of you already studied, solved all the sessions of the IMAT. So there's no point of repeating it with me. So we chose to make our to take from some resources questions that are close to the IMAT, and then we will make them going on. Okay, I think now I can be heard. I'm not knowing what's happening, to be honest. Okay. So we were saying that we're gonna make the PDFs and then we will send them on the website. You go and solve them on the website. They are close to the way IMAT is asked, and they are from Italian books resources. So you might like, it's not like from other kinds of books or other languages. So for the solving, unfortunately, we won't solve together, but we will. So we won't solve together, but we will send them on the website. They will be like 5 PDFs, I guess, and they are new questions, I think. So I think in this all time, I think that you had good enough time to solve the sessions of the IMAT. I want from you in the assignments or in these new PDFs that we will send by today or tomorrow morning. I want you to solve them by time management. How is this? Means you each exam of these that we will send, you have to put 90 minutes trying to solve them, an hour and 40 minutes. And I want you to try your best to be super fast to do it on time. You were not able to do the first one, go to the other. You were not able, means like you finish the first one with the time. You should put yourself under the exam surrounding. You should put yourself under pressure. You should put yourself under the time pressure, under the way you're wearing, the way you feel comfortable. You have to make yourself adapting to the way it will be. Because the main problem of the IMAT is the time management. So now you don't have really good time to study more course. Maybe today is the last day we can speak about course. And today is the last day for the live sessions. However, anything you should do now after this course is make notes. So number one thing after the course, after your notes, I want you to get paper for each section. Let's say biology a paper, chemistry a paper, physics a paper, math a paper. And you write the most important things according to your mind, according to you, the note that you might not remember, the note that you might maybe memorize it now, but forget it tomorrow. So in this way, in each course, in each subject, if you make sure that all the notes that you might not remember on the day of the exam, you wrote them on a paper, on the day of the exam, you will be able to remember them all. Another thing I want to say about this also, okay. So another thing I want to say about this, I really believe that all of you will succeed. If not all of you, will be maybe just one or two. But I think on this hard work that I saw usually during the sessions, on this how we say, the way you were commenting and showing that you know these topics that I'm speaking about already. So I think all of you already have a very good base in everything. And IMAT is usually good base, like for its most basic, but more tricky. It's so tricky. You have to make sure that each question you're solving, you are finding the trick from the question. Each question I'm solving is not a question, oh, just to solve. There's like more than one trick in a question. You have to make sure that you know them. Another thing is that when you solve, start first with biology. First, biology always start, and then later go to the anything else. Other than this, um, try to, let's say you have to know what are the basic or the reactivity of metals or these things, these summaries. Before the exam you will sit like 3 hours or 2 hours before they start the exam. So in these 3 hours, instead of sleeping or speaking to the one next to you, try to remember the sequences you have in your head. Try to make songs in a way to remember everything, all the sequence. So if something came, you already refreshed your mind about it before you start writing. um Hey, even please. I didn't understand. Okay. Other than this, I want to say that for the chemistry part, you mostly you should make sure that you're able to solve fastly. Means you have to practice your hand on the oxidation states. You should practice your hands. So the course must be already inside you, but the most thing you should know is your hand being practiced on the course. Because what think in the IMA, they think, okay, this person already have the course from 3 4 years in his life, and he has been now 3 months just practicing on the course. They don't know that you are studying the course in this 3 months, and you left the practicing for the last month. So the thing you should know is you have to practice as much as possible. Use all these days before the exam for practice. Make your hand work. Write, write, write. Try to balance fast. Try to find the tricks to do things fast. Don't leave yourself in a way, okay, I know the course. I know how is this solved, but I don't know how to do it fast. Because being fast is maybe you know more than the highest one who took in the exam, but the highest one who took in the exam, he is more faster in writing, more faster in thinking, than you are, although you might have more information than him in all subjects. Other than this, in biology and even in all subjects, when you see, especially in biology, but also in all the subjects, when you see a question which is like big question, which looks so big, this means exactly that this question is easy. So don't be scared from the shape of the question because it's big. Try to read the most important things from the question and try to solve it fast. When you see tables, when you see rows, when you see columns, these exact questions are the easiest questions in the IMAP. When you see a question which is a small question, not even a full sentence, this question is the hardest question. Don't be close to it until the end of the exam. Try to solve the easy questions at the beginning. Save your mark and then leave for the end the hard ones. Okay. Okay. So other than this, I want you for the PDFs that we will send. I want you to really, they are not really like, of course, nothing will be higher than the IMAT level, but they might be on the IMAT level or lower. Because usually Italian language level is much easier than, not much easier, but like easier than IMAT level. So we got them and we added on them. So we added some tricks inside the questions. I got the questions from them and I added some tricks on them. So you'll be able at least to find a trick in a question. It's not really direct. The answers will also be given, so you don't have to like do them and send me your work or anything because I know you're already stressed. You need to find the answer. You need to know why the answer is this. You don't really have time for assignments and these things. So what I want from you solve them in 1 hour and 40 minutes, and maybe like solve them in 1 hour and 30 minutes. Don't give yourself much time. In the exam, you will be so stressed, you will be under huge stress, you will see everyone solving, you will see teachers, you will see everyone around you, will see cameras. So all of this will make a huge stress on you. This stress will make you try to make will make you stress so much to not be able to solve. So what will you do here? You will first have to calm down, and then you will have to practice being stressed and being fast in home. So at home, when you're already stressed, when you're already fast, I want you, then you will go to the exam, you will feel it's normal. Normally at home, I die studying the course. I die trying to remember things from the course, like studying and memorizing things. The moment, so then before the exam, by one day, I forget everything. And then I, in my mind, okay, where did everything I studied go? But in the second of the exam, because there's so much stress, you will be able to remember them all in a second. So make sure that the course that you're studying, the course, the memorizing definitions, the sequences, the charts, all of these things that you studied, make sure they are in your mind, stuck in your mind, not really like, oh, I know them and I don't know them. No, make sure that the course is inside your mind. Before you go, the course must be stuck in your mind, so you'll be able to proceed with the procedure of solving of the tricks and of these things. Because the person in the Imat won't be taking higher than 20 or higher than 40 if he just maintains or or focuses on the course and leaves the time management. You must know how to be under stress, you must know how to be solving these things. You must know that this course, they think that you already know the course from grade 12, from the last high school. So much people of you had a gap year. So much of you took like 2 years gap years and aren't really out of high school. So they really think that you already know them from 2 years, and you had 2 full years to practice and be fast. So they think that the course you know it, and the time management you got it in 2 years. They don't know that you didn't know about Italy. You had to study the course again. So this is how Italy works. You have to manage to be like what they think you are. So you get a grade from what they think you might get, right? So please make sure that the course is already in your mind and you're going there just to find the tricks of the questions, not to solve the course. IMAT is not a direct question exam, it's an indirect, super indirect question exam. I'm not making you scared, but you have to put in mind that we are getting close. If you really want to enter medicine, you really have to take into consideration the time management is much important than anything else in the IMAT. Okay. Okay, I want to ask you, did they send you the emails of the test centers? Will you all do it in Pakistan and India, or you will go do it? Is Angela here? No. So where will you do the exam? Is it in Italy, in Pakistan? Where is it? Ah, Pakistan. Oh, you have a center. Oh, really? Lahore. Oh, perfect. Okay, that's so good. That's so good. Some like me in Lebanon, I didn't really have a center in Lebanon. I had to travel to Turkey to sit the exam. So that's so good if you have it already, yani, in your country. Okay, now let's start. India has 3 centers. No way. Okay, that's good. You don't have to work on the visa now, because you should make sure that you will gonna be accepted, then start working on the visa, because the visa papers and the translation really cost so much. 2 centers. Okay. Delhi and Chennai. Okay. So, okay, let's start. So now we will speak about the galvanic cell. I remember I sent on the group what are the things that we should speak about. I'm not finding. Yeah, I found them. So today we will speak about the eyes, the junctions, anatomy, drugs, smoking, naming, and equilibrium and galvanic cell. So what is the voltaic cell or the galvanic cell? The galvanic cell, also known as the voltaic cell, is an electrochemical device that converts chemical energy into electrical energy through a spontaneous redox reaction. It consists of two half cells. Blotting, no, it's not important. They didn't even write it in the syllabus. Biotechnology, we already went through biotechnology, we went through so much things. Did you watch the videos of yesterday? Yesterday was the whole day biotechnology. Okay. So, it consists of two half cells. We have each containing an electrode immersed in an electrolyte solution. The two half cells are connected by a conductive pathway, usually a wire, voltmeter, wire, and a salt bridge. That allows ions flow between the two half cells. In one half cell, oxidation occurs, which is called the anode, and here the electrode. So here in the solution, we have ions. This is the electrode, and then here we have also ions. The salt bridge must be immersed inside. We have here the anode and we have the cathode. The anode is the part where it releases electrons. So electrons come from here to the cathode. Here oxidation reaction happens, and here reduction reaction happens. This conversion of chemical energy to electrical energy, and this movement created, okay, it creates a current, electrical current. The electrode where oxidation occurs is called the anode, while the electrode where reduction occurs is called the cathode. Anode is typically negatively signed, and cathode is typically positively signed. The electrolyte solutions in the two half cells contains ions that facilitate the transfer of charge between the electrodes. Here we have oxidation, here we have reduction. They will end, all this process will end in a redox reaction. And then it will generate a potential difference, which is the voltage. So it will generate a current and a voltage. Galvanic cells are commonly used in batteries and fuel cells. There is mainly a galvanic cell that you might be asked about in the Imat, which is called the Daniell file. So it will have here Zn2 plus ions. It will have in the cathode Cu2 plus ions. So if here we have Zn2 plus ions, means this electrode is Zn electrode. And Cu2 plus means this electrode is Cu electrode. if this is the anode and this is the cathode, so electrons will flow from the anode to the cathode. Perfect. Here we have voltmeter. We have here the salt bridge. Salt bridge will is gonna contain a salt. The salt here is KCl. So usually, since cathode, as we said, is positively charged and anode is negatively charged, so the positive charges will go here and the negative charges will go here. So K plus will go here and Cl minus will go here. Okay, from the salt bridge. And you should know the reactions. We have here the half reaction, oxidation half reaction, it's called. So Zn gives Zn2 plus 2 electrons. Usually in the oxidation reaction, the electrons will be in the product. And however, the reduction reaction, the electrons are in the reactants. So here we start Cu2 plus 2 electrons will give Cu. If we want to find the total redox reaction, it will be like this, the overall reaction, if you want, you can call it. So we have the anode or oxidation half reaction. Zn gives Zn2 plus 2 electrons. And Cu2 plus 2 electrons gonna give us U. Okay, so we take off this 2 and this 2. We have Zn plus Cu2 plus will give Zn2 plus Cu. This is the overall reaction, redox reaction. In the Daniell cell, the anode is made up of zinc, immersed in a solution of zinc sulfate, ZnSO4, while the cathode is made up of copper, Cu, immersed in a solution of copper sulfate, CuSO4. In the Daniell cell, zinc atoms at the anode oxidize, losing 2 electrons, which are transferred through the external circuit to the copper cathode. And at the cathode, copper ions from the copper sulfate solution gain electrons, depositing copper metal on the cathode. This transfer of electrons from the anode to the cathode creates an electric current and potential difference or voltage, allowing the cell to produce electrical energy from chemical energy. Okay. Now, they might ask us, what does the salt bridge help in this redox reaction? So the function of the salt bridge. To neutralize the reaction, to facilitate the flow of ions between the two half cells, okay, to complete the circuit, to prevent mixing of the solutions, to maintain electrical balance, perfect. So these are the functions of the salt bridge. So nothing else you should know about it, Yani. That's only what you should know about, because they really don't need so much information about it. They just need the basic, just to know what is it. Okay, so now, okay, okay, guys. So Sam will speak to you now. He wants to speak to you since it's the last day, and we want to. Today is the last session, so he won't have time later. After like 2 hours, by the
Hello, everyone. It's me. Hello everyone, can you hear me? Amazing. Okay, so I know that Fatima is in the middle of the class now, but I really wanted to take the chance to speak with you guys to make sure where everyone is and if everything is going accordingly, and to explain to you what's going to be happening for the rest of the month. Okay. Um, so first of all, I want to know, actually, at the moment, I'm just finishing like uploading the video to YouTube, basically like showing how to register for the Imat. Was everyone able to register for the Imat, or did anyone have any problems? Okay, that's good. And everyone was able to get their receipt of payment and everything like this, right? Okay, that's good. That's good. Okay. Um, so look, guys, the usually what happened, at least like when I took the IMAT last year, so you get a receipt of payment, obviously saying like that you paid for the exam and etc. Now wait a few days and you should receive like a document saying basically your exam instructions, okay? Like it's gonna tell you what to bring or not to bring and what to expect from the exam date and some basic rules that you have to go over. Um, but basically, what they're going to be sending you is this right here. Let me share it with you. I'm going to share it on the Alpha students. So they're basically going to be sharing this. Wait, this right here. Yeah, so I sent it on a group chat. Yep, just send it. Yeah, so basically they're going to be sending you this. It might be a little bit different from last year. It might not. It might be exactly the same thing, but it should be something like this, basically saying what to do or not to do, what to expect, rules, and that sort of thing. Okay. So whenever you receive this document, make sure you read it very well, because it has very important information. And on the exam day itself, it's very important that you do everything correctly, and you don't really like miss out on marks or miss out on anything just because you didn't know or you didn't read the instructions correctly or you didn't pay attention. Okay. And also like when you know what's going to happen on the exam date, it helps you kind of like prepare a bit more, give you a peace of mind. So now that's that. Uh, the second thing is that it's really beneficial. I've been speaking with some of you guys on private already. Now, something that is going to prepare you mentally, because look, the exam is very stressful. A lot of people stress so much for the exam. Okay. And sometimes you might actually be perfectly prepared for the exam, but just because you're stressing for the exam, you might get a very bad grade because you're not really able to analyze everything. You're not able to um, process everything because you're just so nervous and you're so worried that you'll get the right answer and so worried if everything's gonna go well and because this might be your last chance or Allahu Alam, right? Um, So it's really important that you guys start preparing your psychological state for the IMAT as of now. Okay. Now, how can you do that? So one of the ways that you can do that, and I highly suggest, is that you start to take every single day. I would say minimum 3 times a week. Wake up at 8, right? Have a similar schedule that you're going to have on the exam date, right? Let's say if the exam date, you're going to wake up at 8, right? You're going to eat, you're going to get ready, you're going to travel to the exam center. Do something similar. Wake up at the same time. Um, Eat, etc. Do your little revision before. And then at 12, exactly Italian time, you're going to go and do your exam, okay? Sit down in a room. There's no one in a room and sit down with a paper and pen, or maybe if you don't have paper and pen, you can just use your phone and then put the answers down on a sheet of paper. Put your phone on airplane mode, just download the IMAT test paper and just basically solve it. Okay. Because this is basically going to prepare you with timekeeping. It's going to prepare you to get used to the exam itself because a lot of the times the IMAT exam is something so new for everyone. Like we're like a lot of people are not really prepared, they're not really knowing what's going to happen. And that fear of the unknown actually impacts a lot of people in a very negative way, which is not good. Um, so yeah, I really suggest doing that. Okay, really, really important. Every day, try to, at least 3 times a week, try to put yourself in the same situation um as of the IMAT that you would do in the IMAT exam. Okay, does that make sense, everyone? Does that make sense? Cool. So now I want to give some tips as well for when you're taking the exam itself. Whenever you're gonna take the exam itself, really the first thing that you should do, you should guarantee your free easy marks. Okay. Now the free easy marks is usually logical reasoning, critical thinking. Okay, like that first section. Okay, usually they are the easy marks. And by the way, like I'm basically inshallah today I'm gonna be uploading the full like videos on every topic of the critical thinking and logical reasoning. And so you guys can have access to that as well, inshallah. Um, but yeah, those are the easy marks. So you actually want to go to the entire paper, right? And find the easiest marks and do them first. Okay. You should why? Why is this important? Because one, you're gonna start the exam. In the next 5 to 10 minutes, you're already gonna get so much marks. So much marks. And that actually is going to make you very like positive. That's gonna make you less stressed. That's gonna make you like be more positive about the exam and therefore your outcomes is going to be much better. Okay. And obviously if you took the time to solve the easy questions first, you're going to have enough time to solve the hard questions later. Okay, so this is an absolutely important thing. Don't try to do anything else. Don't jump straight to the math section. Let me do the hard thing. Don't do that. I'm telling you, you're gonna regret it. Do not do that. Just go and solve all of the easy questions. Go to critical thinking, go to biology, go to every single question, and the easy ones that you see, solve all of them. Then once you go from cover to cover and check every page and then all of the easy questions, I would say you should minimum be already having 20 points. Minimum, like that's minimum, right? Because a
Yeah,
lot of the questions
exactly. And
they're very
I'm
easy.
sorry, I have to say something. If you feel that any question that you didn't understand or you didn't or you're not sure to, even if you know about the topic, even if, or let's say you know all the course but you forgot something that will make you like let's say 90% unsure about your answer, don't put the answer, leave it empty, don't put the answer. However, if you felt you have like a 95%, it might be 95%, put the answer which you think it's right. Trust when you are solving, trust your answer, don't trust another person's answer, or don't trust the let's say, oh, it came in last year's session and the answer was like this. No, trust your answer. You already have the full knowledge, you took the whole course, you have the full knowledge, you are thinking as the professor is thinking. So anything that you feel after, of course, after you found the trick, after you solved, after you did everything, make sure that your answer is the correct one. Don't at the end of the this last 5 minutes say no, I will change it. Don't change your first answer, the first answer that you took in the first, in this question. Oh, you said, oh, I think it's this one, put it this one. Don't change it at the last 5 minutes. This is a huge mistake that most of the students do.
100%. And in case, let's say if there's a question that you're very unsure, just like mark it, leave it to the end, right? And then maybe like after you solve all of the questions, and then when you're basically at towards the end, maybe your memory already refreshed and you might actually have the answer for the question. So this is very important. Are you guys familiar how the IMAT answer sheets look like? Please like put in the chat so I can know. Okay, are you guys familiar with the format? How does it look like, etc.? Uh, you're not okay. Cool. I'm gonna open WhatsApp. I'm gonna send you exactly how it looks like, so you guys know what I'll be talking about next. Okay. Um, one sec. Okay, let's see. Okay, everyone, please try to go to the WhatsApp group. I sent the picture, so you guys know what I'm talking about. Okay, perfect. Right. So this is the IMAT answer sheets, right? So now, obviously, like the IMAT past papers that you guys download online to solve, etc. You're not gonna be writing on them. I mean, like you can write on them, but you're not gonna be putting your answers on them, right? And let's say if you did everything correctly, but you put it your answers on that paper, guess what? You're gonna get zero. Okay, you're gonna get zero. You won't get nothing. So it's really important that while you're, let's say if you want to highlight something or underline it or whatever, that's cool. But whenever you have the answer for that specific question, you go to the answer sheet, which is a separate piece of paper, okay? And then in the answer sheet, you go and you mark, right? Let's say option A, B, C, or D, or E, right? Then you mark which one it is, just so you don't forget, because a lot of students, they end up forgetting or they end up marking on the other paper, the one that you're supposed to just do the reading, and actually they don't get the mark, you know? And make sure that even after, let's say, you solve all your exam, make sure you double check that the answers that you put on the answer sheets matches exactly with the IMAT paper, okay? You do not want to be losing marks for something silly like this. And it's really important that you're checking back and forth and making sure that everything is in the way that it's supposed to be. So yeah, obviously you can write it. You can write stuff on the IMAT paper, but just don't forget to put your answers on the answer sheet. Is that clear? Does anyone have any questions about this? You on the question paper, yes, but you cannot like I don't suggest writing on the answer sheet, but you can write on the question paper. Um, yeah, in case you guys didn't see it, this will look like. Does that make sense, guys? Do you guys have any questions about that? In return, question papers will open marks. If you mean, Said, if we have to write, if we have to put on the answer sheets on pen, yes, obviously you have to put pen. It cannot be pencil, it cannot be nothing. And plus, they're gonna give you a pen. Okay, you cannot use nothing else except a pen. And by the way, like, I don't know if you guys know, but let's say if you, I don't know, let's say if you mark something wrong. Let's say if you want to, if let's say if you want to go back to your answer, if you want to choose something else, you can. Usually, like, they're going to send you a document on your email in the next following days, basically talking to you what you have to do in case, let's say, you chose a wrong option. You tell the one that is correct and like fill in the whole box on the one that is incorrect. Okay, but don't worry about that. Don't stress about it. You're gonna get an email explaining everything. Okay. No, 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 no. They will accept, bro. Like they do not care. Understand? They don't care about the question paper. They don't care about it, right? What they care is the answer sheets. The answer sheets, as long as the answer sheet is good, they don't care about the question paper itself. You won't get no grades. It doesn't matter. They don't care about it, right? It's just for you to read, basically. But what really matters is your answer sheet, the one that I send the picture on. That's all that matters. Nothing else. Does that make sense? Guys, look, if you have any questions specifically about the how is it going to be on the exam date and how is the whole thing is going to happen, what type of papers you're going to get, and Like, feel free to ask. Don't be shy. Like, here's not the place to be shy. Just let me know. Okay, cool. I assume that everything makes sense since no one is saying anything. Um, okay. Do we have to stay behind after the exam? Okay, so look, I'm going to if we get the sequence wrong in the answer sheet, bro. That's what I'm telling you, right, Saeed. Rafi, I'm gonna come back to you in a sec. Look, if you get the sequence wrong in the answer sheet, this is why you should be checking, right? Let's say if you wrote it in the wrong way or that's why you should make sure to review what you put it. Let's say after you finish solving the exam, make sure that you putting the right thing, right? And then whenever you're writing on the answer sheet, look at the answer sheet. Say, okay, this is question number 5, and the correct answer is B. Then you cross check and make sure that it matches. Okay, it's really important that you make sure that you're cross checking everything. Okay, now let me explain how is it going to work on the exam day itself. So you're going to arrive at the exam center. You're going to put all your backpacks, your stuff, your watches, and everything like on a specific place. Some of them give you some bags that you can put it inside. Some of them they just give you like a place that you can put your things at. Okay, you take all of your things out, put over there. Now you won't have nothing with you, right? And then they're gonna obviously like ask your name and etc. You're gonna have to show them like your registration from the Imat. Okay, sorry guys. So yeah, so you're going to have to show them like your receipt of payment. Then when they see you in the system, then you can go in a little line where you're gonna enter the exam room. Okay, so everyone sits on the exam room, right? And then they're going to explain to you the rules of the exam. They're going to say this is what you can do, this is what you cannot do. The exam is going to be X 100 minutes, right? Um, and whenever you finish, all you have to do, you have to walk in and give your paper, basically. Okay? No, no, sorry. So yeah, so the beginning, you're gonna sit down, and then the invigilator is gonna come and give you the papers, okay? And then they're gonna tell you when you can start writing on it. You cannot start writing immediately. You have to wait until they allow you to write. Then they'll say, okay, now you can write your name. Now you can write your student number and whatever, you know. And then after this, they'll tell you, okay, you can start your exam. Then you start your exam, and then after you finish your exam, after the 100 minutes, right? They're gonna say, okay, now everyone pen down. If you start writing, they'll kick your ass out, right? Don't write. When they tell you stop writing, you stop writing. Put the pens down. Halas, done, right? You did what you can. When they tell you write, you write. When they tell you don't write, don't write. Just listen to them, because if anyone thinks that you might be cheating, I'm telling you, they will take you out of the exam and you're done. You're finished. There's no buys, there's no cries, there's no nothing. They won't care. Okay, so just do what they're telling you to do. So yeah, so you finish the 100 minutes, then they're going to start collecting. And they're going to start. One second. Okay, so yeah. So then basically like they're gonna tell each student like from, let's say if there's 10 rows, like rows of students, right? Then they're gonna say, okay, row 1, get up and give the paper. And then that's how it is. You just go up, give the paper, put there at least for me there was a box that you put your answer sheets, there was a box that you put the the questions, but don't worry guys. I'm just letting you know just so you're a bit familiar with it. But
So on the box that you have to put the answer ones, and on the box that you have to put the question, it's there is a paper which is printed on the box, the shape of the paper that you hold in your hands. So you have to look just at the box, you will see the print, and then you will put it inside. Your big folder, the big one that contains all of these papers, you should throw it in the box which is on the chair, which will be right next to the desk. The desk will have these two papers. On the paper, which is the paper of when you write your name and when you sign. First, you have to write your name, your birth date, and where the place that you're born. Means you write, let's say India, and you write the place where your village is. After this, you don't. It will have a place where you have to sign. You don't sign it. You leave it for the end. You go to the desk when it's your turn, and then you take, because when it's your turn, they won't allow no one to explain for you what you should do. You should do it by yourself. You do it wrong, you did it wrong. You take a zero. So they will explain it at the beginning of the exam. At the end of the exam, no one will explain nothing for you. You cannot ask for help for any person in the place. So what you should do, you take your two papers. There will be and the folder. You put the folder. You will have two papers: the paper where you wrote your name, the paper of your answers, and a third, and all other papers, which are the question sheet papers, the explanation papers, all of these things. The question sheet and the explanation, you put it inside the folder. The folder, you put it on the chair in the big box. There will be two small boxes. The two small boxes will have a print on them. You will see how the shape of the paper looks like. You put the shape of it, and then you put it inside the paper, which you have, which is your answer sheet. You don't have to write your name on it. You just have to put your answers and put it in the box. The other paper will have two stamps. You should put sticky things that you should put on, which is a code. This code, you choose it. You will have two codes. You will have two codes on it. You will use one code for the above part of the paper, and one code for the bottom. I guess you should put on the answer sheet in the Imat. I don't know if it's the same of the Italian language. So you have to make sure if there's a place where you can put a code on the answer sheet. After this, you don't have to memorize the code. So much people will tell you, oh, try to memorize it in the anonymous results. You will know. Even if you try to memorize it, you won't be able to memorize it. You'll be out of stress. You want just to go home. Don't stress yourself. Don't memorize it. It won't help you nothing. Even
Yeah,
if you knew it, so much people will be like, okay, 677. There's thousands and thousands of numbers like this. So don't even stress yourself and memorize things which won't help you nothing.
exactly.
So after this, you will stick the code. After you stick the code, you have to sign. Down there is a place for signature. You sign after you finished your exam. This sign recognizes that you did your exam, not someone else. After you finish your signing, you put the paper and you can say bye, you finish. Okay. So thank you so much, Sam, for
Mhm.
coming. And I think now everything is so. Thank you for helping me and explaining the way the exam is. And I think now everything is clear for you guys. If you have any questions to Sam, just tell him before we continue our
Yeah,
session.
guys, and by the way, don't worry about this. We're just like letting you know now, just so your brain start like getting used to the exam format, just so you psychologically you're ready. But obviously, like don't worry. Like a few days before the exam, we're gonna do our best to try to look for the papers and to explain it in a bit better way, for you to be ready and everything. But don't stress. The whole point of us telling you all of this is just for you to be ready and not to worry about it so much and use your stress just to learn and practice the IMET, which you need to do. And yeah, guys, that's pretty much it. And
Okay, now,
sorry, just one
yeah.
more thing. And yes, we are working for the what's it called يعني, for the MCQs and the practice papers, and inshallah we're gonna be submitting them on
Yeah,
the
we will send the
time
practice
assessments.
papers by tomorrow. Tomorrow or after tomorrow, maximum after tomorrow. They will be on the website. There are new questions other than the IMAT sessions. I think you're done with the IMAT sessions. So there are like new questions. You can test yourself on them.
Exactly, 100%. Okay, good. I will let you guys finish the class then. Thank you very much. Anything, just message me on the
Okay.
IMET of WhatsApp.
Okay.
Thank you.
So now we're gonna speak about the physiology of the eye. So let me screen share. So, so here the anatomy of the eyes. This is the eye. We have the number one thing outside is the conjunctiva, which is this layer here. And then we have the cornea. Then we have the anterior chamber. Then the iris. Okay. So conjunctiva, ciliary body, the iris, the anterior chamber, the cornea, the pupil, the lens, the posterior chamber, the canal of sclem, the conjunctiva from the down side. This conjunctiva is a kind of layer from up to down. The vitreous, the sclera, the choroid, the retina, the macula, artery, central retinal optic nerve, the vein, which is the central retinal vein, and the rectus medialis. Okay, so conjunctiva, ora serrata, ciliary body, aqueous, iris, anterior chamber, posterior chamber, cornea, pupil lens, canal of sclem, conjunctiva, from down, vitreous, sclera, choroid, retina, macula, artery and vein, optic nerve, and rectus medialis. Okay, what is the human eye? The human eye is a complex organ responsible for vision. It works by receiving light, converting it into electrical signals, and transmitting these signals to the brain for interpretation. The physiology of the eye involves various structures, each playing a specific role in the visual process. Let's speak about the external structures before we start on these internal structures that we just spoke about. What the eyelids, what is their function? They protect the eye from debris, regulate light entry, and spread tears across the surface to keep it moist. The eyelashes, they help to filter out the dust and debris. The lacrimal glands, they produce tears which lubricate the eyes and contain enzymes that protect against infection. The eyeball structure. The eyeball consists of three layers: the outer fibrous layer, the middle vascular layer, and the inner neural layer. The outer fibrous layer, it contains the cornea and the sclera. The cornea is the transparent dome-shaped front part of the eye that refracts light entering the eye. However, the sclera, okay, is the white part of the eye made of dense connective tissue, provides structural support and protection. The middle vascular layer, or the uvea, the iris, is the colored part of the eye surrounding the pupil. It controls the amount of light entering the eye by adjusting the size of the pupil. Okay? Pupil. The black circular opening in the center of the iris that regulates the amount of light that enters the eye. Ciliary body contains the ciliary muscle, which changes the shape of the lens for focusing like accommodation, and the ciliary processes which produce aqueous humor. The choroid is a vascular layer between the sclera and the retina, providing oxygen and nutrients to the eyes. The inner neural layer, which is the retina, which all of it is nerves. We have the retina is a light sensitive layer containing photoreceptors, the rods and the cones that convert light into electrical signals. What are the rods? Rods are photoreceptors sensitive to low light levels, providing black and white vision. The cones are photoreceptors responsible for color vision and sharpness in bright light. The macula is a small central area in the retina responsible for high acuity vision. Fovea centralis is the center of the macula with the highest density of cones, providing the sharpest vision. Optic disc, the point where the optic nerve exits the retina, creating a blind spot since it contains no photoreceptors. So in the optic disc, there is no photoreceptors, and the retina, we have the photoreceptors, which are the rods and the cones. Optic nerve transmits visual information from the retina to the brain. What are the internal fluids in the eyeball? The aqueous humor and the vitreous humor. The aqueous is the clear fluid produced by the ciliary body filling the anterior chamber between the cornea and lens and maintaining intraocular pressure while providing nutrients to the lens and cornea. However, the vitreous humor is a gel-like substance filling the posterior chamber between the lens and the retina that helps maintain the eye's shape and supports the retina. So the aqueous chamber, the aqueous humor, it helps in maintaining the intraocular pressure and the nutrients. However, the vitreous chamber helps maintaining the shape of the eye and support for the retina. The lens is a transparent, flexible structure behind the pupil that focuses light onto the retina. The shape of the lens is altered by the ciliary muscles to focus light from different distances onto the retina. Light pathway. What happens? How is the light entering? So let's share. Yeah. Okay, so let's do this. So let's speak if the light enters this eyes, the eyeball. What happens to this light? Where does it go? What are the steps it focuses on to reach the end of the eye? Is it charging? Yeah. Perfect. So let's speak about the process of this light. So light enters the eye through the cornea. So one is the cornea. I'm sorry guys, someone is calling me for a room. Okay. So the light pathway goes from the cornea. Number one thing is the cornea. So the light enters the eye through the cornea, passes through the aqueous humor, then to the pupil, then to the lens, then to the retina. Cornea, aqueous humor, pupil, lens, retina. The cornea and lens, they refract light to focus it to the retina. So these two, they refract light to focus it on the retina. The iris adjusts the pupil size to control the amount of light entering. So, who is the main reason of the size of the pupil? Is the iris. The iris chooses what size of the pupil it must be for this amount of light entering. Let's say we have a very high amount of light entering entering this eye. The iris will immediately make the shape of the pupil like this. However, if there is very low amount of light, the pupil will becomes bigger and bigger like this. Why? Because when you have so much light, you really don't need so much effort to see. However, or space to see. However, if there is not really good amount of light entering your pupil, you really need a very huge amount of space for this light to start entering more from the pupil. Who's the main reason of this? Who's the alarm of this is the iris. Okay. This contraction and relaxation process is called the accommodation process. Okay. The biggest example is the cat. You see the cats at morning, their eyes look like this. At night, they become like this. Because at night, there's not really this huge amount of light, so they really need space to see more light. Okay. Okay, so we said that the lens and the cornea, they refract the light. The iris, it adjusts the pupil size. The lens changes shape to focus light on the fovea for sharp vision. Photoreceptors, the rods and the cones, detect light and convert it into electrical signals. Bipolar cells and ganglion cells process these signals, which are transmitted from the optic nerves to the brain. So when it reaches the retina, it will reach the optic nerve. This optic nerve will then take it to the brain by the help of um, the translation of this message by the help of the bipolar cells and the ganglion cells. Now let's speak about the neural pathways. Optic nerve. Optic nerve carries signals from each eye to the brain. Okay. Optic chiasma. The point where the optic nerves from each eye partially cross, ensuring visual information from each visual field is processed by the opposite hemisphere of the brain. Means let's say we have here the right eye and here the left eye. Means the optic nerve of the right eye, the optic nerves of the left eye. And here is the right brain, and here is the left brain. What will happen? The right eye will take this message and take it to the left brain. And the left eye will take the message to the right brain. This is called the optic chiasma. Okay? Optic tracts continue from the optic chiasma to the lateral geniculate nucleus and the thalamus. Lateral geniculate nucleus, so in the thalamus we have the LGN, the lateral geniculate nucleus. This optic chiasma, after this message is took to the brain, we have inside the brain, we have this thalamus. It will reach in the brain the exact part which is called the thalamus. From this nucleus inside the thalamus, a relay station in the thalamus that processes visual information before sending it to the visual cortex. So here it will reach the nucleus and then it will go from the nucleus to the visual cortex, which is also in the brain in the occipital lobe. If you remember, we spoke about the occipital lobe. So it takes it to the visual cortex. and then the information are processed, allowing us to perceive the images. Okay. Now let's speak about visual processing. So, perception of light and color, rods and cones. These are in the retina, the photoreceptors. They respond to different wavelengths of light. Cones are responsible for color vision with three types: small, medium, large, that detect short, medium, and long wavelengths respectively. So cones, okay. So cones is responsible for color vision from its name, color vision. Three types: small, medium, large. They detect short or medium or long wavelength. Short means the blue light, medium means the green light, and long means the red light. So they see all of these. Okay. Image formation. The brain interprets the electrical signals from the optic nerve to form a coherent image, taking it into account light intensity, color, depth, and movement. Accommodation, the process by which the lens changes shape to focus on objects at varying distances, controlled by the ciliary muscles. So let's write the definition of the accommodation. Pupillary light reflex, the automatic constriction of the pupil in response to bright light, which protects the retina from excessive light exposure. Okay. Now let's try to do a comparison between the rods and the cones. What are the rods? What are the cones? Rods are low light vision, means this scotopic vision, means low light vision. Rods are highly sensitive to light. And allow us to see in dim lighting conditions. Means help us to see in at night. They help us to see in the black and white vision. They do not detect color. And peripheral vision. Cones are color vision, are daylight vision. Color vision, central vision, and rods are concentrated on the peripheral part of the nerves of the eyes. However, the cones are concentrated on the central part of the nerves of the eyes. So rods have a long cylindrical shape with a stacked arrangement of membrane discs containing the photopigment rhodopsin. So rods are made up of rhodopsin. The pigment of rods is rhodopsin. They look like this. However, however, the cons looks like this, the same as the rod shape, but each one of them have a color because they detect colors. Rhodopsin is a highly sensitive to low light levels, is the primary photopigment in rods. Rods respond more slowly to light stimuli compared to cones. Cones are shorter and tapered to a point resembling a cone shape. Okay, the cones contain three pigments. Here we have the rhodopsin, here we have the opsins. Okay, opsins, they work on these three types. We have the red cones, or let's say the L cones, the M cones, and the S cones. These small ones are called the opsins. However, the small part, the unit of the rods are the rhodopsin. So rods are predominantly located in the peripheral regions of the retina, and they are absent in the fovea. So you should memorize this note that they're absent in the fovea. Cones are concentrated in the central retina, and they are in the fovea. Because the fovea is basically the center of the retina. So since rods, they prefer to sit on the sides of the retina, on the peripheral parts, and the cones chose to sit inside. So in the fovea, we have cones on the center, and the peripheral part of the retina, we have rods. What else you should know? That rods are slow to get a response of this light. However, cones are fast. The number of rods in the retina are around 120 million, and the number of cones in the retina are around 6 million. So we have more rods than cones. Okay. Okay. Okay, perfect. So we're done with the eyes. I don't think you should know more than this. That's a brief explanation about the eyes. Now, let's speak about junctions. So, we have different types of, let's speak about junctions of animals, junctions of plants, junctions of the body. It's not charging. Let me just make sure it's not charging. Yeah, it's charging. Okay. Let's see if someone asked. Ciliary muscles. No, ciliary muscles are around the iris. When you contract your eyes, when your eyes contract, ciliary muscles relax. When your eyes relax, ciliary muscles contract. Let's say here is your eyes. Here are the ciliary muscles. Okay. And then the ciliary muscles, they contract when they see so much lights. You know, when you see a very high, let's say you're used to sit in, let's say you're at night, okay, you're sleeping, and then you woke up and you opened your eyes. When you open your eyes won't be open perfect. Why? Because it's contracting, it's closing. So it's the main reason of this contraction because it's very high light for you because your body is not adapted for high light in this amount of time. So that's why like your eyes will close immediately. This ciliary muscle is it's a muscle, so its main function is contracting and relaxing the eye. Okay. Yeah. Okay. So cell junctions in animals and the humans, and cell junctions in plants. We have the tight junctions. We have the adherence junctions or the belt desmosomes. We have the macula adherenza or adherence. We have the gap junctions, which are called also the communicating junctions. We have the hemidesmosomes. Okay. However, the junctions in plants, we have the plasmodesmata. We have the middle lamella. Mm, I think that's all. Okay. So let's think about each one of these junctions. The tight junctions, the occluding junctions. Tight junctions are composed of transmembrane proteins such as claudins and occludins that seal adjacent cells together. These junctions form a barrier that prevents the movement of substances between cells, particularly in epithelial tissues. This helps maintain distinct compartments within the body by regulating the passage of molecules and ions. So the main function of the tight junctions is forming a barrier that prevents the movement of substances between the cells. Okay? The location is in epithelial cells lining organs like the intestines, bladder, and the blood-brain barrier. So main location is epithelial cells. Adherence junctions or the belt desmosomes, they composed of cadherin proteins. However, the tight junctions are made up of claudin protein and the occludin. So here we have claudin protein and occludin. However, the adherence is made up of cadherin. Okay. which are linked to the actin cytoskeleton within the cell. These junctions form a belt-like structure around the cell. Their function is provide strong mechanical attachments between adjacent cells, means they stick two cells together, helping tissues resist mechanical stress. Their location are also epithelial cells, such as the intestine lining and the skin. The macula adherents or the desmosomes are formed by cadarins, like the since macular adherens and the adherents normal, the belt ones, both of them are desmosomes. Like the desmogleins and desmocollins, connected to intermediate filaments like keratin. So the adherents, the belt ones, are connected to actin. However, the macular adherents, these type of desmosomes, they are connected to keratin. They provide strong adhesion between cells, particularly in tissues that experience significant mechanical stress, found in tissues such as skin, heart, and muscles. So these are found in tissues. Gap junctions or communicating junctions composed of connexin proteins that form the channels connectons between adjacent cells. Gap junctions allow for direct communication between cells by permitting the passage of ions, nutrients, and small molecules, which is essential for coordinated cellular activities. The gap junctions are found in cardiac muscle cells, in smooth muscle cells, and in certain neurons. So gap junctions in the cardiac, smooth, and neurons. Hemidesmosomes. Hemidesmosomes. They are similar to desmosomes but connect cells to the extracellular matrix rather than to other cells. They are composed to integrins that anchor intermediate filaments to the basal lamina. So the protein is called the integrin protein. They connect to things, the intermediate filaments to the basal lamina. Hemidesmosomes anchor epithelial cells to the underlying basement membrane, providing structural stability. They are found in epithelial cells, particularly where they attach to the basement membrane, such as the skin. Okay, now let's speak about the cell junctions in plants. Plasmodesmata are channels that traverse the cell walls of plant cells. So here they exist in the cell walls, consisting of a narrow tube of endoplasmic reticulum called the desmotubule surrounded by cytoplasm. So they are consisting of a tube of endoplasmic reticulum called the desmotubule. Yeah. And this tube is surrounded by cytoplasm. These junctions allow direct communication between plant cells by enabling the passage of ions, signaling molecules, nutrients, and other small molecules, thus maintaining tissue cohesion and coordination. Plasmodesmata are ubiquitous in plant tissues, particularly in areas where cells must work in concert, such as in vascular tissues like the xylem and the phloem, and during growth and development. The middle lamella is a pectin-rich layer, so it's rich in pectin. That glues adjacent plant cells together, found between the primary cell walls of adjacent cells. It provides structural integrity and adhesion between cells, maintaining the overall structure of plant tissues. Found in all plant tissues, especially in those that require strong intracellular adhesion, such as the parenchyma. Okay, so these are different junctions you must know. You must know tight junctions, adherence junctions, macular adherence, gap junctions, hemidesmosomes, plasmodesmata, middle lamella, and that's all. Okay, now let's speak about drugs and smoking. Drugs are substances, so drugs are substances that have physiological effects when introduced into the body. Types of drugs are stimulants and depressants. Stimulants are like cocaine, amphetamines, amphetamine, caffeine, and nicotine. The effect of the stimulants, they increase alertness, they increase energy, they increase heart rate. In high doses, they can cause anxiety, they can um, yeah, they reduce appetite. The person would not want to eat anymore. They can cause paranoia. Paranoia means anxiety and boredom, and they can cause heart problems. Depressants are like alcohol, benzodiazepines, barbiturates, opioids, heroin, morphine, Valium. These depressants, they slow down brain function. They reduce anxiety. They induce sleep. They can cause respiratory depression. They can cause coma. They can cause death. Okay. So we spoke about the stimulants and the depressants. Let's speak about the hallucinogens drugs. Hallucinogens are like LSD, are like psilocybin, MDMA, PCP. These hallucinogens, after perception, mood, and thought processes, they can cause hallucinations and distortions in time and reality. The person who takes these hallucinogens, they will start to hallucinate in what time is he, in what is it real, is he in a dream, and like this. Okay. We have the opioids, drugs, like heroin and morphine and fentanyl, oxycodone. These have a powerful pain relief. They can cause euphoria, they can cause drowsiness, they can cause respiratory failure at high overdose, of course. So, this is all you should know about drugs. I think you should just know that they end up with toxification of the body, and you should just know that liver tries as much as possible to get rid of these toxins of these drugs by breaking them down. However, so much kinds of drugs are not able to be really detoxified by the liver, leading to problems in the body and the heart and even in the liver. Because high amounts of toxic things that enter the body, the more the liver will work. The more the liver will work, the more the more toxic, the more the liver will work, the more it might not give a good function, and it will end up in a liver failure. Let's speak about smoking. Okay, so let's speak about the health risks of smoking. Smoking, like tobacco, everything, inhaling smoking, and the burning tobacco smoking can lead to serious and death problems. Tobacco, what are the components, which is nicotine, tar, carbon monoxide, and other chemicals. You should know that carbon monoxide itself is really toxic. Hemoglobin in the body is able to absorb carbon monoxide 200 times more than oxygen. So if you will give your body carbon monoxide, your blood will is gonna absorb this carbon monoxide much more easier than oxygen. Will lead to lung problems, to breathing problems, to asthma, to COPD. All of these problems are the respiratory problems. The main reason of them is the carbon monoxide, which is found in tobacco smoking. So, smoking might lead to cardiovascular diseases, They might lead to stroke. They might lead to respiratory diseases, chronic diseases like COPD, bronchitis, emphysema, okay, damaging of the alveoli. They might lead to cancer in the esophagus, cancer in the bladder, cancer in the pancreas, cancer in the lungs, cancer in the mouth, cancer in the throat. They might lead to reproductive problems. It might lead to infertility in both men and women. And it increases the sudden infant death syndrome, the SIDS. And it might lead to immune system suppression. All of this because of high heart rate, high blood pressure, and that's why all of these happens. Okay? This is for the first-hand smoker. Second-hand smoker, the smoke exhaled by a smoker or emitted from the burning end of the cigarette, which is involuntarily inhaled by others. Second-hand smoker has the higher ability of having these problems, of having these symptoms than the smoker himself. Because the smoker himself is getting this carbon monoxide at the same time he's taking out a very good amount of carbon monoxide from his body by exhaling. However, the secondhand smoker is the person who sits next to the smoker, is the one who's getting highly destroyed by this smoking because he is inhaling it and he's not letting it out in any way. So these symptoms might appear in the second-hand smoker much higher than the first-hand smoker. Okay. No, I don't think so. You should know the respirometer. Okay. Now we have to speak about the organic reactions that we have in our body. Organic reactions involve chemical processes that take place between organic compounds. Organic reactions are fundamental to the synthesis and transformation of biomolecules. Okay, so we have substitution reaction. We have addition reaction. We have also oxidation reactions, reductions. I have elimination reaction. Substitution reaction is like we substitute a compound from and to reach another compound. Let's say R minus X plus Y gives R minus Y plus X. We substituted their places. In a substitution reaction, one atom or group of atoms in a molecule is replaced by another atom or group. We have two types of substitution reaction. We have the nucleophilic substitution and we have the electrophilic substitution. A nucleophile. What is a nucleophile? Nucleophiles are usually the electron-rich species. The electrophile are okay. Electrophiles are the complete opposite. Are the electron less species, poor species. Replaces the leaving group a halogen. Let's give an example of the substitution reaction. Let's go here. CH3Br plus OH minus gives CH3OH plus Br minus. We substituted the place of the Br and the hydroxide ions and we ended up in this reaction. However, the electrophilic substitution, this about the nucleophilic substitution. However, the electrophilic substitution is C6H6 plus NO2 plus gives C6H5NO2 plus H plus. So what is the electrophilic substitution? An electrophile, electron deficient species, replace a hydrogen atom in an aromatic ring. So here we substituted this NO2 with one hydrogen here. Okay. In this fourth substitution, let's speak about the addition. Addition reaction means means B plus C Addition reactions involve the breaking of a double or triple bond in a molecule and the addition of new atoms or groups. We have also the electrophilic addition and the nucleophilic addition. Means when you have a double bond and then you break this double bond because double bond is usually unsaturated. So you can break and you can add. Break this double bond and the scarcity. We end up in single bonds. Elimination reactions. Elimination reactions look like this. Involve the removal of atoms or groups from a molecule resulting in the formation of double or triple bond. Let's take an example. Elimination is forming from a single bond a double bond. This is the complete opposite of the addition reaction. Redox reactions, we already spoke about redox reactions. Rearrangement reactions. Rearrangement reactions are the structure of a molecule is rearranged to form an isomer where the connectivity of atoms is changed. Means let's say we have a butene, it will become a butene in the presence of an acid. Say the catalyst is an acid. This rearrangement, what did we arrange here? We just rearranged the double bond functional group. So this is called rearrangement reaction. We have condensation reactions. Condensation reactions involve the combination of two molecules with the elimination of a small molecule, usually it's water. Let's say we have a carboxylic acid and we have an alcohol. Okay, we end up having and water. So here when they add together, this OH will go with one H here. They will end up with water, and then we will add this R prime group or this radical to here from the carboxylic acid. Okay, this is called a condensation reaction. Polymerization reaction involve the repeating linking of monomer units to form a larger polymer. So this polymerization is also a kind of from small molecules to big molecules. And we have the esterification reaction, which is this one. This is the esterification reaction. And ending up with an ester using a carboxylic acid and using an alcohol, ending up with an ester and water. From esterification or condensation reaction, esterification, if we want to start with an ester and end up with a carboxylic acid and the alcohol, we have to do the complete opposite. If you remember when we first started speaking about the types of reactions we have, we said we have two kinds of reactions. We have the dehydration or condensation and the hydrolysis. So what's the opposite of this? The opposite is hydrolysis reaction. What is hydrolysis reaction? Is the complete opposite of this, starting with an ester and ending up with one alcohol and carboxylic acid. These are the different types of the organic compounds, organic reactions. Okay. Okay, now we see what we still have. Speak about naming organic compounds. What do you mean? Is it exothermic? Which reaction you mean? Okay. Just give me 1 second. I'm seeing what do we still have other than naming, because I remember I sent on the group yesterday everything we still have. So I want just to check them. Where is it? If anyone, okay. You just put a dot. Eyes, junctions, anatomy. Yeah, we still have anatomy. Drugs, smoking, equilibrium, naming. So we still have equilibrium, naming, and anatomy. Okay. So what was what were we speaking? So condensation reaction, confused because I read anabolism, and condensation is exothermic. Condensation can be both, can be endothermic and exothermic. Okay. Let's say if you're speaking about esterification, here heat is released. Who can see what? Okay. So if you speak about acidification, it's exothermic. Okay, but there are some exceptions which it can be endothermic. Let's say we are having, like, how is it endothermic? If the bonds are being broken stronger than the bonds being formed, and or if the reaction involves overcoming significant activation energy, here it's endothermic. However, acidification is exothermic because we are releasing heat. But there are, yeah, same goes for hydrolysis. Both of them, because they are completely, like, they are the opposite of each other. So both of them can be endothermic and exothermic. However, if the bonds which are being broken are stronger than the bonds being formed, it's endothermic. And if the opposite, it's exothermic. And if the reaction is overcoming activation energy, it's endothermic. If the reaction is releasing this activation energy, it's exothermic. And what I want to say is that when we spoke about anabolism and catabolism, we said that catabolism usually is the decomposition. Decomposition means it's releasing heat. You are decomposing something, so it will release heat while decomposing. Let's say you have carbohydrates. When you decompose, when you break down this carbohydrate, it's already storing heat inside, so this heat will be released. So yeah, it's exothermic. Anabolism is endothermic. Okay, now we want to speak about how to name organic compounds, or nomenclature, or the IUPAC name. So we have, of course, like 1 methyl, 2 ethyl, 3 propyl, or let's say methane, ethane, propane, butane, pentane, hexane, heptane, octane, nonane, decane. Okay. The carbon atoms in the parent chain, the carbon atoms in the parent chain are numbered to give the lowest possible numbers to the substituents or functional groups. If multiple substituents are present, the chain is numbered from the end closest to the first substituent. Let's say we have so what's the name of this? 1, 2, 3, 4, 5, 6. We don't stop here. We try to do other ways. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. No, 5, 6. So the longest chain is this chain. So we start by counting the longest chain. And then we start from the closest. Here the branch is closest to here. It's far from here. So we use this as 1, 2, 3. Okay, so it's 7. So it ends in heptane. It does not have double bond, so it's ane. And we have here on the third, okay, so 1, 2, 3. So 3, say this are methyl. We have 2 methyls, so 3, 3, and we have another methyl here on the 4. So 3, 3, 4, trimethyl heptane. Di means 2, tri means 3, and 1 means mono. Alkane usually single bond, alkene usually double bond, alkyne usually triple bond, alcohol have OH. Aldehyde CHO. Carboxylic acid. Ester. Amine. Um, amide. Nitrile. Nitrile. Triple bond with N. And ketone. That's all. So to name the alkene, let's take an example. We have methane. To name the alkene, ethene. To name the alkyne, ethine. To name the alcohol, ethanol. To name the aldehyde, ethanal, Al. In the aldehyde, you end it up with Al. To name the ketone, propanone. To name the acid, the carboxylic acid, ethanoic acid. Oic acid. To name the amine, let's give an example. Ethylamine. So it ends up with the same amine. Amide, ethanamide. Nitrile, ethylnitrile. Okay, these are the things. What else? If let's speak about halogens, let's say we want to name with the halogens. What are the halogens? Bromine, chlorine, iodine, fluorine, fluoro, ethane, iodio, ethane, chloro, propane, bromo, methane. Okay, so bromo, chloro, iodio, fluoro. Okay, what else? So, for naming, it's not really hard, it's easy. First, you have to take the longest chain, and then you have to see the branches, and then you have to see if we have double bonds, and then you have to make sure if we have any acyl group. Usually methane is CH3 mostly used, ethane is CH5, CH5, okay, or CH3, CH2, CH3, ethyl, methyl, and yeah, I think that's all. If you want to solve, you have so much books, you can solve from them. Of course, if you want to speak about the double bond, let's say we have this. It will be 2 ethene. No, this ethene. Let's say we have this. Okay, so 1, 2, 3, 4, 5, 6. So hexene, but we have it on 2. This one, this. So we start counting from the closest part of the functional group. Functional group is most close to this side, so we count from here the 1. Okay, so it can be to hexene or hextuene. Goes to you, what you choose. It's the same. And what else? Go over organic isomerism, structure chain. Yeah, isomers. Okay. Okay, so there are different types of isomers. We have the structural isomers, or called also the constitutional isomers. we have the stereoisomers, we have we spoke about them, positional isomers, functional isomers. So structural isomers, they can be chain isomers or also called the skeletal isomers. They differ in the arrangement of the carbon chain. Okay, let's say we have two isomers. Let's say we have the butane this is the butane, and let's say we have mm, isobutane or 2-methyl propane. Okay, okay, it can also look like this or like this. This is called the iso. Butane, everything iso means it has a shape of a tree around a carbon. So this is also called the 2-methyl propane or the isobutane. So isobutane, okay, and the 2-methyl propane, these are called the chain isomers or the skeletal isomers. Why? Because they are the same exact shape. And the butane, they have the same formula. Let's say 1, 2, 3, 4 carbons, 3, 4, 5, 6, 7, 8, 9, 10, 10 hydrogens. Here we have 3, 4, 5, 6, 7, 6, 7, 8, 9, 10, also here and also here. So all of them have the same molecular formula, but different structure. This is called the chain isomers. Okay. Ring isomers are also considered chain isomers. Functional isomers. Functional group isomers differ in the function, but they have the same molecular formula, and they differ in their just in the function, I guess. Means we have alcohol and ether. Both of them have the same molecular formula, but they differ in the functional group. Alcohol has the hydroxyl functional group. The ether have the O, the OR R prime functional group. Okay, or okay, let's say we have ethanol and dimethyl ether. Okay, so both of them have the same function, same molecular formula, but different functional group. Here we have this group, and here we have the alcohol group. Ring chain isomers are structural isomers. So ring chain isomers, they differ between the linear and the cyclic forms. Let's say we have the cyclohexane and hexane. Both of them are the same, but Both of them are the same, but their structure is different. Cyclohexane looks circular, and the hexane looks linear. We have the tautomers are also structural isomers, so tautometric isomers. They are rapidly interconvertible forms of the same compound, usually differing in position of a hydrogen atom and a double bond. Let's say we have the ketoenol, like acetone. Acetone is usually in ketone form, and its enol form, which is the propanol. Prop, propanol. Okay. So these are called the tautomeric isomers. These tautomeric isomers they happen usually between, they differ usually in the position of the hydrogen atom and the double bond. So they have double bonds, both of them. But the position of the OH here is different than the position of the OH here. This is called the tautomeric isomers. Okay. Lastly, we have to speak about the positional isomers. Positional isomers, they differ in the position of the functional group. Let's say we have one butanol and two butanol. Here we have OH. Here we have the OH. Here it's on the one, here on the second carbon. Here's first carbon, here's the second carbon. Here it's on the first carbon, here it's on the second carbon. The functional group is the place of the functional group is different in the number of carbons, and that's why we have the same molecular formula but different structural formula. Stereoisomers, which are the last kind of isomers. We have the geometric isomers like the cis and the trans. They differ in the spatial arrangement of groups around the double bond or ring system. Let's say we have the butene. It can be a cis butene or a trans butene. If these two, if both of them, or if both of the groups are on the same side, this is called the cis butene. However, if the groups are on the opposite sides, this is called the trans butene. Let's try to draw it. Okay. Let's say here we have H here, H here, CH3 here, CH3 here. CH3 and H here. Here we have CH3 up and H down, so this is called the transbutene. However, if we have CH3 here and CH3 here, and H here and H here, both of them on the sides, both of them on the same side, this is called the cis butene. Other than this, we have the enantiomers. They are non-superimposable mirror images of each other, usually due to the presence of a chiral center, a carbon atom bonded to four different groups. Like the lactic acid, it has two enantiomers. Lactic acid has the L-lactic acid and the D-lactic acid. Okay, so we have a chiral center bonded to four different groups. We have, other than the enantiomers, we have the diastereomers, stereoisomers that are not mirror images. These are mirror images. You know how you put an organic compound like this, and you put it on the mirror, and then it will show in this way. This is called enantiomers. Like they show the same opposite on the mirror. Their stereomers are not mirror images of each other. They have different configuration at one or more chiral center, but not all. Here we have a chiral center has four groups. Here we don't have a chiral center. In a molecule with two chiral centers, such as tartaric acid, like the mesoform and two non-mesoforms, these are called the diastereomers. The last kind of stereoisomers are the conformational isomers or the rotamers. They differ in the rotation around single bonds. Okay? These are not true isomers, but they represent different conformations of the same molecule. We have the shapes, which are the rotamers. You will take them next year, inshallah, if you got accepted in medicine. You will take them in details, the staggered ones, the eclipsed ones, and each energy ones. You know, so these conformations or the rotamers, they just differ in the rotation. Let's say you have a molecule looks like this, and then it will start rotating. It will start going up, and then start rotating. And the way it's rotating, of course, does not look this easy, but it's much harder to make resonance while rotating and then to reach this shape, which is the most stable shape in nature. Okay, so it will keep on rotating until it reaches a very stable shape, which with a good amount of energy to with a less energy to be able to stay in nature. Okay. Uh, yeah, positional, functional, and chain are all structural isomers, exactly. And ring isomers also. Let me tell you what are the structural isomers or constitutional isomers. They are chain isomers or skeletal isomers, positional isomers, functional isomers, ring chain isomers, tautomeric isomers. These are the structural. And we have the stereoisomers like the geometric isomers, like the cis and the trans, optical isomers, diastereomers, conformational isomers. Okay. Now, let's speak about the Le Chatelier's principle. Okay. Let's say we have any kind of reaction, which looks like this. So, if you see here in this reaction, it's an equilibrium reaction. Means it can go in the right position, it can go in the left position. Depending on what? It depends on the concentration of the reactants. It depends on the pressure. It depends on the temperature. And yeah. So if we said that in which direction will the equilibrium move if we added more A, if we added more concentration of A, the equilibrium will start moving from the most concentration to the less concentration. So the equilibrium will start moving from here to here. If we said that we heated up this reaction, means we added heat, we must first take into consideration delta H, which is the enthalpy. If delta H is negative, this means that this reaction is exothermic. However, if delta H is positive, this means that the reaction is endothermic. If delta H was negative, this means that here is the heat, is in the product. So then the reaction will go from where is the heat. So it will go from here to reduce the heat. If delta H is positive, this means that the heat is with the reactants. It's endothermic, it needs heat. So from where the heat here is here, so it will go in this direction. If we increase the pressure, Le Chatelier's principle tells us that go and see where do we have higher number of molecules in the reaction. The equilibrium will shift from where the high number of molecules is there. Okay. So here we have A and B. Here we have how many molecules? We have two molecules. However, here we have AB acts as one molecule. So the equilibrium will shift from the more molecules to the less molecules, means from here to here. So pressure depends on molecules. Temperature depends on delta H and heat. Concentration of reactants, so easy, where you see high concentration, it will go in its direction. Okay. Another note, at equilibrium, the rate of the forward reaction is equal to the rate of the backwards reaction. Okay. What are the factors that can change the rate or that can increase the rate? Concentration of the reactants, catalyst or catalysm, and temperature. No, wait. Let me make sure of the temperature. 1 second. Does the temperature, because yeah. So yeah. It can only if the Le Chatelier's principle exists in this. Okay. So yeah, that's all. What you also should know about equilibrium. You should know the constant of equilibrium constant. So we take the coefficient, we put it as a power, and we multiply the concentration of the products over the concentration of the reactants. This is the equilibrium constant, and yeah. What else you should know for today? Still have, I guess, only anatomy. Okay. Okay. KSP is a solubility constant. Kc and Kp is Kp is the pressure constant, Kc is the concentration constant. So if you speak about pressure, you put Kp. If you speak about concentration, you put Kc, and the solubility constant is the Ksp. Ksp is usually only the products, only the reactants with their coefficients. Okay. Okay, let me write it. Okay. Um, Rate or velocity if we add base, water, or acid. You mean if we add the hydroxide ions and H to the equilibrium, you mean, or to the whole reaction? What do you mean by if we add acid and base? I remember we used to draw. You're speaking more about acid base. You speak about more about the pH where it reaches 7. When you put strong base. When you put weak base with strong acid. I know what you mean. I think to be honest, these I didn't study them for the Imat because they won't like you won't have time to draw a chart and to put n-1, n+1 to end up finding the x and these things. I wanted to teach you these, but in my mind I was like maybe like it's not really asked, you know? Like they won't ask you this deep. It's more about super basic, you know? However, like adding an acid increases the concentration of H plus ions in the solution, and it might affect equilibrium, which are more basic. Let's say we have let's take an example. Let's say I have an H3 plus H plus gives an H4 plus. Okay. If I added an HCl, means I'm adding H3O plus or H plus. Means I'm adding more acid. Here I will increase the concentration of H plus ions. So if I increase the concentration of H plus ions, what happens if you remember we said if we increase the concentration of the reactants, Le Chatelier's principle takes the reaction to the direction to lessen this concentration. So it will go this way. Why? Because we're getting from the high concentration to low concentration. Let's take another example. Let's say I have H plus plus OH minus gives H2O. If I added a base, NaOH, means I'm adding OH minus. So I'm increasing OH minus, so the equilibrium will shift from here to here. Even if I add H plus, it will also shift from here to here. Um, yeah, so it's just like a kind of way to make sure that what are we adding acids or bases and which direction according to the Lachatelier's principle. Okay, you should not really know this topic to be honest, even if they wrote it in the syllabus. It's not really asked. It might be asked, but not really this much. Okay, now let me screen share. So here we have the skeletal system and the muscular system. We have here the frontal lobe, we have the temporal lobe, we have the nasal, not lobe, I mean the skull, the front of the skull, the temporal skull, the orbit, the nasal, the maxilla, the mandible, cervical vertebra, clavicle, true ribs, humerus, false ribs, radius, ulna, carpals, metacarpals, phalanges, public symphysis, the sacrum, the coccyx, the ilium, the floating rib, the lumbar, the, the xiphoid, the costal cartilage, sternum, and the ischium, the pubis. The femur is the biggest bone, patella, tibia, fibula, talus, phalanges, metatarsus. However, in the muscular system, we have the rotator cuff, the biceps brachii, the brachialis, the pronator teres, the brachiodialis, iliopsoas, abdominal external oblique, rectus abdominis, pectoralis major, the deltoid, the sternocleidomastoid, orbicularis oculi, peroneus longus, quadriceps femoris, adductor muscles, tibialis anterior, and the peroneus brevis, and that's all the muscles. You should focus on the skeleton more than the muscle. Okay. So, clavicle, sternum is in the middle of the ribs, pelvic girdle, the ulna, the carpals, metacarpals, phalanges, femur, patella, tibia, fibula and tibia are together in the legs, metatarsals, scapula is right under the clavicle, the ilium, the ischium, and the pubis, the clavicle and the sternum and the skull. Let's go to the skull. We have the zygomatic arc, zygomatic bone, the maxilla, the mandibles, the occipital bone, temporal bone, parietal bone. They are the same of the lobes, right? The frontal view, we have the nasal bone, the vomit, the mandibles, the maxilla, lacrimal bone, temporal bone, sphenoid bone, and the ethmoid bone, the ethmoid and the sphenoid. and inferior nasal concha. Okay. Now I want to give an additional note about the anatomy. So if you saw that all of these bones are all connected to each others, only there's only one free bone in our skeletal system which is not connected to the whole skeleton, which is called the hyoid bone. The hyoid bone is here, is right under the tongue, which is here. It's not written here. Why? Because it's not connected to them. It's called the hyoid, and it's between the throat and the upper jaws. This hyoid bone is a free bone and it's not connected to our bones, to our skeletal system. Okay, I guess these you should try to find an application to memorize them. What else? Humerus is one of the biggest bones of the hands. And yeah, that's all I guess for today. We finished today, we finished the course. I guess even if we didn't go through others, I guess you can take them or you can go through the other things using practice in the 17 days. And inshallah, we will speak to you before the exam by some days. We will make another meeting. We will prepare you. I will make a note paper. Um, we'll go around like notes, like hard things you might forget. I will tell you about them. I'll make a note paper and we will explain it together in the session right before the exam. Now have a good night and bye bye.